Welcome to the video on computer-aided drug delivery systems, specifically Unit 2, Part 3, covering computational modeling of drug disposition. This lecture is presented by Dr. Krishna Yadav.
Computational modeling of drug disposition is defined as a quantitative framework for predicting and simulating ADME processes, that is, absorption, distribution, metabolism, and excretion. It uses mathematical, statistical, and mechanistic models to understand drug behavior. Instead of relying only on experimental trials, we simulate drug movement across the body. Think of it like pre-formulation prediction. Just as we evaluate solubility or stability before formulation, here we evaluate in vivo fate before clinical studies. This improves efficiency, reduces cost, and enhances rational drug design. Computational modeling of drug disposition comprise computational modeling of drug disposition, focusing on the ADME processes, absorption, distribution, metabolism, and excretion. Starting with drug absorption modeling, it considers dissolution, solubility, permeability, and intestinal physiology. This determines how much drug becomes available for systemic circulation. Next, drug distribution modeling describes how the drug moves into tissues, reflecting tissue uptake and binding. Then, drug excretion modeling explains how drugs are eliminated via renal and biliary routes. An important addition is active transport in drug disposition, where transporters significantly influence absorption, distribution, and elimination. Under absorption, we further consider solubility modeling, determining drug availability; intestinal permeation, drug transport across intestinal epithelium. Under active transport, efflux transporters pump drugs out, reducing absorption. Uptake transporters facilitate entry into cells. Specialized transporters handle specific molecules, like peptides.
Active transport profoundly affects drug disposition, influencing absorption, distribution, and elimination. Transporters are vital for predicting bioavailability and drug-drug interactions. We categorize them as efflux, uptake, and specialized transporters, based on their function and role. Having understood transporter classification, we now focus on transporters. Efflux transporters, like bouncers, pump drugs out, restricting CNS penetration. Uptake transporters, the importers, facilitate drug entry. Specialized transporters are substrate selective, crucial for targeted drug delivery. Efflux transporters pump drugs out, limiting absorption and protecting cells, while uptake transporters facilitate cellular entry, critical for hepatic uptake and renal secretion. Both systems are governed by Michaelis-Menten kinetics, described by the equation shown here. The Michaelis-Menten equation, J equals J max times C over Km plus C, describes the transport rate, where J max is the maximum capacity, C is the substrate concentration, and Km, the Michaelis constant, defines the affinity for the drug. This slide summarizes the major transporters controlling systemic drug exposure. In the gut, PGP and BCRP pump drugs back into the lumen, reducing the fraction absorbed, Fu. In the liver, OATP 1B1 over 1B3 and OCT1 enable drug uptake, while PGP-BCRP support biliary excretion, making the liver a key control point for metabolism and clearance. In the kidneys, OCT2 drives renal secretion of cationic drugs like metformin. At the blood-brain barrier, PGP and BCRP act as protective gates, limiting CNS drug entry. All these processes combine in the equation F equals Fu times Fg times Fh, showing how transporters directly regulate systemic exposure. Overall, this works like a multi-step biological drug delivery system, controlling how much drug finally reaches circulation. Key transporters include PGP and BCRP for efflux, limiting oral bioavailability and CNS penetration. October and OATP are uptake transporters in the liver and kidney, mediating cationic and anionic drug transport, respectively, and are important in drug-drug interactions. P-glycoprotein, or PGP, is found in the intestine, liver, kidney, and BBB. It pumps substrates out of cells, reducing oral absorption and CNS penetration. BCRP shares functional overlap with PGP and is a critical determinant of oral bioavailability and CNS exposure. P-glycoprotein, or PGP, encoded by the ABCB1 gene, is an ATP-dependent efflux pump. It plays a key role in restricting drug absorption while promoting drug elimination. Mechanistically, PGP actively transports structurally diverse drugs out of cells using ATP. This process follows saturable kinetics, described by the equation shown, where transport depends on Jmax, Km, and substrate concentration. Looking at its locations, in the intestine, it pumps drugs back into the lumen, reducing oral bioavailability. At the blood-brain barrier, it restricts CNS penetration, acting as a protective gate. In the liver, it supports biliary excretion. In the kidney, it contributes to renal secretion. Pharmaceutically, PGP behaves like a biological barrier system, actively reducing the effective drug concentration available for therapeutic action. PGP's clinical significance lies in protecting the body from foreign substances. Its activity impacts drug disposition and therapeutic outcomes, contributing to drug resistance and drug-drug interactions. It is a key determinant of bioavailability and CNS penetration. Breast cancer resistance protein, or BCRP, encoded by the ABCG2 gene, is an ATP-dependent efflux transporter that influences drug absorption, distribution, and elimination. Mechanistically, BCRP actively transports a wide range of substrates out of cells using ATP hydrolysis, and it shows functional overlap with PGP. Its transport follows saturable kinetics, as described by the equation shown. Looking at its locations. In the intestine, it limits oral absorption, affecting bioavailability. At the blood-brain barrier, it restricts CNS entry, protecting brain tissue. In the liver and kidney, it contributes to biliary and renal excretion. In the placenta, it acts as a protective barrier for the fetus. Pharmaceutically, BCRP functions like an additional biological barrier layer, further reducing drug availability and influencing therapeutic outcomes. BCRP's activity is crucial in protecting the body from foreign substances and has significant implications for drug pharmacokinetics and toxicity. It is a key factor in oral bioavailability, CNS protection, and multi-drug resistance. Genetic variations in ABCG2 can alter BCRP function. In summary, P-glycoprotein and BCRP are major efflux transporters. Both are ATP-dependent. PGP, encoded by ABCB1, limits oral bioavailability and restricts CNS penetration, contributing to multi-drug resistance. BCRP, encoded by ABCG2, also limits bioavailability and CNS penetration and acts as a protective fetal barrier. Uptake transporters include organic cation transporters, or OCTs, and organic anion transporting polypeptides, or OATPs. OCTs transport positively charged drugs and are critical for renal tubular secretion. OATPs mediate the hepatic uptake of anionic drugs and directly affect first-pass metabolism. Organic cation transporters, or OCTs, belong to the SLC22A solute carrier family and are responsible for the cellular uptake of organic cations, including many drugs and endogenous compounds. They play a key role in drug disposition, particularly in hepatic uptake and renal tubular secretion, thereby influencing systemic exposure and therapeutic outcomes. Mechanistically, OCTs transport small hydrophilic cations across membranes via electrochemical gradients without direct ATP consumption. Their kinetics follow J, Jmax, and Km, similar to carrier-mediated systems. Looking at the key isoforms, OCT1, SLC22A1 is predominantly in the liver, mediating hepatic uptake of orally administered drugs. OCT2, SLC22A2, is highly expressed in the kidney, critical for renal secretion of cationic drugs. OCT3, SLC22A3, is widely distributed across multiple tissues, contributing to diverse physiological roles. Pharmaceutically, OCTs act like drug entry facilitators, enhancing intracellular drug availability at target organs. OCTs are significant determinants of drug pharmacokinetics. OCT1 mediates hepatic clearance, while OCT2 facilitates renal tubular secretion of cationic drugs. October inhibition or induction can cause drug-drug interactions, altering drug exposure and therapeutic effects. Organic anion transporting polypeptides, or OATPs, belong to the SLCO gene family and act as important influx transporters responsible for the cellular uptake of a wide range of endogenous compounds and drugs. They are especially significant in the liver, where they mediate the initial step of hepatic drug elimination, particularly for anionic and neutral drugs. Mechanistically, OATPs facilitate Na-independent transport of large, hydrophobic organic anions. This process involves exchange with intracellular anions, allowing substrates to move along their electrochemical gradient. Their kinetics follow J, Jmax, and Km, similar to carrier-mediated uptake systems. Key isoforms include OATP1B1, SLCO1B1, highly expressed in hepatocytes, mediating uptake of statins, bilirubin, and bile acids. OATP1B3, SLCO1B3, liver-specific, involved in uptake of statins, bile acids, and peptide drugs. OATP2B1, SLCO2B1, more broadly expressed, contributing to diverse drug disposition pathways. Pharmaceutically, OATPs act like targeted entry gates, ensuring drugs reach the liver for metabolism and clearance. OATPs are major determinants of drug pharmacokinetics. OATP1B1 and OATP1B3 are key for hepatic drug uptake, especially of statins, and their inhibition can lead to drug-drug interactions, increasing systemic exposure and the risk of myopathy. Genetic polymorphisms in SLCO1B1 affect drug response. This diagram summarizes the key drug transporters, including PGP, BCRP, OCTs, and OATPs, illustrating their locations and roles in drug absorption, distribution, metabolism, and excretion throughout the body. The references used in this video include key texts and articles on clinical pharmacokinetics, PBPK modeling, and drug-drug interactions involving metabolizing enzymes and transporters. They also include documentation from SimSip and GastroPlus simulators.